Welcome to AP Biology. Today we're talking about analyzing experimental design, and I have some practice questions for you. Let's do a quick run through of these vocab words first. Independent variable is what I decide. So if I'm the experimenter, I decide what I want to do, and this is what I set up. So this is what I know before I even start the experiment. The dependent variable is what you measure, so it's the results really of the experiment. Constants are everything that you need to keep the same. We sometimes call those controlled variables, not to be confused with a control group. So these are anything that you're going to keep the same through the entire experiment. We'll do examples of that as we go. Okay, and then you have experimental groups, at least one, usually more, and then control groups. You've probably only encountered negative controls up until this point. That's what AP mainly is concerned about, but once or twice I've seen positive controls. So we'll define what each of those means as we go through these examples. So control group. First of all, AP Central loves to ask about control groups. FRQ means free response questions. On a regular AP Bio test, there'll be two big questions that will take, I think it's 20 minutes each, and then there'll be four other questions that are shorter. Unless I don't know what they'll do this year. Last year, they only did two questions total. But anyway, either way, one of the big FRQs is always on an experiment. So often they want you to set up your own experiment, or sometimes they might ask you to analyze an experiment that's already been done. They almost always ask about the control group. What would be a better control? Why was this the control? What is a control? So you should, if they ask that, you should tell them what a control is, that it's a baseline for comparison to the experimental group. And then you should say what the control in this particular example is. Okay, so when they're talking about control groups, they usually mean a negative control. And this is really what you've been doing probably through all of your science courses up to this point. So this means that they want you to pull out one of the variables in the experiment in order to have a baseline for comparison. So in a negative control, you expect nothing to happen. So in this example up here, you've got some plants and you're watering them, and then you've got some plants and you don't water them. So this would be the control group, and this would be the experimental group, and the experiment might be what's the effect of water on plant growth. And so in this case, you've pulled out the water, so that's why this one is your control group. And it gives you the baseline. If the control group grows anyway, then you'll know that you don't really need to water plants. They didn't grow because they got watered. Okay, so one thing that I think makes an AP class expectations a little bit different is that in the past, you might have just gone with regular conditions and said, hey, that's my control. So for example, let's say you're doing an enzyme experiment and you're testing your enzymes. Let's say you're testing your enzymes at pH 3, and then you have an enzyme reaction. So what you would need is your reactant, and you'd put that in your test tube, and then you'd put your enzyme in your test tube, and then hopefully that would make some kind of a product. And I know we haven't covered this yet, but you put that stuff in here and you make it pH 3, and you say, hey, how much product is made. We could do that in another test tube at pH 5. We could have another one that's pH 7, another one that's pH 9, let's say, and then let's do pH 11. And they might say, hey, what's the control group here? Or is there a control group already? And up until this point, you probably would have just said pH 7. That's the normal one. That's what we're going to compare them to. And I mean, you're kind of right. That is probably what we're going to compare them all to. But that's not a control, because maybe the experiment works better at pH 5. I don't know. It depends on the enzyme and what pH it works best in, right? So for a control, they actually want you to pull something out. So you would need to have another test tube, and you would want to put no reactant in it at all, or maybe no enzyme. You would have to pull something out. So if you put no reactant in this one and you just put the enzyme in here and you still saw a product made, then there's something totally messed up with your experiment. So for a negative control, you pull something out of the experiment that acts as a baseline for comparing everything else to, and you expect to have no result. Another good example of a control in this experiment would be putting in the reactant but no enzyme. If you did that, you still shouldn't expect product to be made. Okay, so here's the key. Don't just go with regular conditions. So don't say it's pH 7, that's my control. Don't go with like, daytime or 37 degrees Celsius, like 37 degrees Celsius is body temp. You actually need to remove something in almost all cases in order to have a control group. So we'll go through a bunch of, I have like 10 examples or something. We'll go through a bunch of examples so that that makes sense to you. Okay, so if you have to explain the function of a control group in an experiment, you should explain that it's used as a comparison or a baseline to the experimental group. And you can even go into a little more detail. So in this case, they're not saying, what is the control for this experiment? They're saying, maybe what is the control for the experiment, but also what is a control? Why is this a valid control? And you should explain that it's used as a comparison or as a baseline with which to compare to your experimental group. And you could even go further saying that if like for example with the enzyme one, if I still got a product produced even though I didn't put in any enzyme, I would know that my experiment was had some kind of a problem with it. Okay, so here's an example from part of an AP question. It says identify a control group from the missing experiment and justify the need for this control group in the experiment. So they often ask about experiments and they'll say identify the control group that's missing because this experiment doesn't have a control group, and then justify the need for it in this particular case. So we'll go through some examples of that. Okay, so the missing control is needed because the effect of the independent variable and the dependent variable, right, the effect of water on plant growth, can't be determined without the comparison of the plant growth without the water, right, under all the other same conditions, the same amount of sunlight, same type of soil, et cetera, et cetera. So the missing control is needed because the effect of X on Y can't be determined without comparison to Y under the same conditions without X. Fill in your X and Y. So for this particular case, the missing control is needed because the effect of water on plants can't be determined without the comparison of plants without the water. All right, here's another example. I don't really know how to say this. This is an experiment that we actually do in class if we get together. It's guaiacol, I think. It's an indicator, and actually indicator is something really, really important that I want you to know. So an indicator indicates something. Often it changes color. So this is something that turns brown when it reacts with oxygen. So if you have guaiacol, I don't really know how to say that. Anyway, if you have guaiacol around in a stoppered bottle, it will be clear. And if you open it up and mix it with oxygen, or if you have maybe photosynthesis going on and it's producing oxygen, then it's going to turn brown. Or any other reaction, it'll turn brown. So here's a chemical reaction. We've got hydrogen peroxide, and you put it with this enzyme, peroxidase, which breaks down hydrogen peroxide into water and oxygen. So in this experiment, we would have some test tubes, and in the test tubes, we would put hydrogen peroxide, and then we would put peroxidase, and we would also put the guaiacol in it. It would start out clear, but as this reaction proceeds to the right, it's going to make oxygen, which will bind with this indicator, and it'll turn brown. That indicates how much of the reaction has happened because it indicates how much oxygen is produced. So it says here, the browner a sample becomes, the more light it absorbs. So we actually end up putting these into these cute little cuvettes, which look, instead of like a test tube, it looks like this little clear, like a square tube. And then they can fit into this machine that we have. And so you have your whole reaction going on in this. And then you put it into a little machine that sends light through, and that measures absorption. So A389 just means it's measuring the absorption at that particular wavelength. So the browner it is, the more light will be absorbed. If this is totally clear, all the light's going to go right through, nothing gets absorbed. If it's really dark, if it's dark brown, then most of the light is going to go here, but it's not going to be able to get through. It'll be absorbed. So let's look. So there are 5 test tubes, and there are all these different pHs. So this is showing a change in absorption after incubation with peroxidase. So they put in this cuvette, they put in the hydrogen peroxide, this stuff. They put in peroxidase, and they put in guaiacol, this right here. And then the browner it becomes, the more oxygen there is, which means the more the reaction has happened. So if you look at these numbers, it looks like the biggest one is right here at pH 7. So that's saying at pH 7, the enzyme works most efficiently, the reaction goes faster, and I know that because it's getting browner. More and more light is being absorbed because this is brown, and so light can't be going through it. So that means more light's absorbed because it's getting darker. It's getting darker because the guaiacol is combining with the oxygen, making everything brown. Okay, so hopefully we'll do a lab with absorption and indicators, so that will help drive it home a little bit. Propose an appropriate control treatment to the experiment below and describe how the control would increase the validity of the results. Most of my students at this point said, oh, pH 7 is a great control. Yay, all done. It's great because it's like the normal pH. So yeah, it is the neutral one, right? Same amount of acid and base, which makes it neutral, right? But that's not what they're looking for. They want to have something pulled out of the experiment so you can compare the experiment to it and make sure that the experiment's actually running appropriately. So what they're looking for is for you to say that we have another test tube 6, and we don't put hydrogen peroxide in it. And if there's no hydrogen peroxide, you shouldn't be making any oxygen. You would just have, like, the hydrogen peroxide would normally be in water. So you would just have some water without hydrogen peroxide, and it shouldn't be turning brown, right? You'll put here's your cuvette. You'll put some water in here because the hydrogen peroxide is actually dissolved in water. And then you'll put the peroxidase in here too, and it won't turn brown. So that would be a nice control. Or you could put in the hydrogen peroxide solution and not put in peroxidase. So either one of those things, you would expect to have no oxygen produced, and then that means it won't be combining with the guaiacol, and you shouldn't turn brown at all. It should be very clear. And so that would be a nice comparison group. So I don't know if I listed everything, but for the control, maybe you would put in water and peroxidase, and you would have to put in that indicator. I can't spell it. AIACOL. Yay. Or instead of putting in the peroxidase, you could put hydrogen peroxide in instead. Okay. Okay, and then it also says, describe how the control would increase the validity of the results. If the solution still turned brown with no hydrogen peroxide, then there's something else going on here because that shouldn't have happened. Or if the reaction still turns brown without peroxidase around, that means the reaction is still happening. I don't know what's going on here, right? That would help you describe how the control would increase the validity of the results. Okay, example number 2. This is one that we actually looked at in our last video, but I only sort of just showed you the numbers instead. And so now we're going to actually talk about the experiment a little bit more. So there's an investigation of a particular insecticide. An insecticide, this is something that kills stuff. In this case, it kills insects, right? Fungicide would kill fungi. Bactericide would kill bacteria, right? Regicide kills the king, right? So an insecticide is something that kills insects. So what's the effect of this particular insecticide on plant growth? Well, that's weird because the insecticide kills insects, but do the insects help the plant or do the insects hurt the plant? So are these insects that kill and eat plants, or are they insects that maybe pollinate plants? Ooh, I don't know yet. Let's read. So on plant development, researchers established 20 test plots out in nature somewhere containing mature Haplopappus squarosus shrubs. I looked them up. They look like this. Aren't they pretty? 10 test plots were sprayed with water-soluble insecticide. This is actually kind of important. Water-soluble insecticide. That means when you have your spray bottle and you put your insecticide in this spray bottle, right, the thing that kills the insects. Oh my goodness. Let's draw a little nozzle here. I so struggle with the drawing part. Okay, so here's my lousy nozzle. Not only do you have the insecticide in here, but it says right here that it's water soluble, and that's actually a really important thing to notice. But there's also water in here. So you've got water and you have insecticide. So it turns out that little 2-word thing, water soluble, becomes important in this question. I'll show you why later. But it means it's not just like a powder or something, there's water in it. Okay, so 10 test plots were sprayed with water-soluble insecticide and 10 test plots were sprayed with water only. Researchers recorded the number of plants or plant parts at different life stages in each test plot. So how many flowers do you have? Well, for water spray, there were 8,000. I'll show you what this means later. For insecticide, there were 9,000 and plus some more. You waited to see how many seeds were made. There were 2,000 and something seeds made in the plots that had water, and more of them, 3,000 in the insecticide. Ooh, there's more with the insecticide around. So I bet that means that these insects, well, this thingy is killing insects and the plants do better. So the insects must be eating the plants, right? At the seed maturation level, there are 60 for water spray and 1,200 for insecticide. So you end up, the insecticide helps the plants to make more seeds, make more seedlings, because that's seedling development here. Okay, so here are the results. Then they ask you what the control group is, and they ask why the researcher didn't just use unsprayed plants as the control. So you are supposed to say that the control is the water spray, right? So you have 20 plots of these shrubs that you spray with water, and 20 parts that you spray with the insecticide. So this is the experimental group, and this is the control group. My students were actually really good last year at coming up with the control group. What they didn't do very well with was this part about the why. So it says, explain why the researcher didn't just use unsprayed plants. And like 90-something% of my kids said, well, plants die without water. Duh, which is totally true. They do. But that's not what they were looking for. So a control pulls out one factor, right? The control will pull out one factor from an experiment, not two factors. And so this is a water-soluble insecticide, which means you're spraying the plants not only with an insecticide, but also with water. If you had another plot that you were not spraying at all, they wouldn't even be getting the water. So you would be taking out two things, right? Not one. You would be taking out the insecticide and you would be taking out the water. And you're really only supposed to take out one thing. So why did the researcher not use unsprayed plants? Well, because that would be taking out two factors, insecticide and water. So be careful. They're almost always expecting you to say, for the control group, we're pulling something out. We want to only pull out one thing and keep everything else exactly the same. So we can say, yeah, it was the insecticide that affected the plants, not the water. All right, and that's where I explain the water-soluble thing. Okay. Okay, here's another one, control group example 3. So they give you a whole thing to read about this, and so I'm just going to describe it. So there are like little fruit flies that got little wings and buggy eyes and legs, and you put them in here. And so let's say you put like 20 little fruit flies in there, and some of them will kind of walk or fly over here, and some of them will walk and fly over here. And this chamber is maybe made out of clear plastic, so you can look inside, and it has a top to it, and it just has that one little hole right here. And they guess maybe they put their finger over it to make sure or something over it to make sure the bugs don't escape. So then they wait a certain amount of time, and they see how many of these fruit flies end up on the glucose-soaked cotton over here, and how many end up on the dry cotton over here. And if they all come over here, or if a lot of them come over here, right, it's sort of random, but maybe they can sense the glucose, maybe they can smell it. If a lot of them end up over here, then we can say, oh, these fruit flies really are attracted to the glucose because it's a food source, yum, yum, right? So it says, propose one specific improvement to each of the following parts of the experimental design and explain how the modification will affect the experiment. So the experimental control, what they are hoping that you'll see is that this glucose-soaked cotton has not only sugar, right, not only glucose, but also water. So this dry cotton has two things removed. That's not okay. It has to have one thing removed. So if you soaked this cotton in water instead, now we would know if the fruit flies come over here that they're sensing the glucose, not the water. Then we would know that the fruit flies can sense glucose, right? And then environmental factors, explain how modification of environmental factors will help. So you come up with like whatever is reasonable here. Like maybe we have the same amount of sunlight in each side. Each side should be the same temperature. It should have the same, I don't know, It should be physically the same size. I guess they kind of already are, but really anything reasonable that you can come up with there is good. Like make sure the cotton is the same size. Make sure it's the same amount of cotton. I don't know. Come up with whatever you can and explain it, and you probably won't get points off there. Okay, let's do one more. Control group number 4. All right, so here's another example. Lipase, you might remember ACE means it's an enzyme. Lipase, ooh, it breaks down lipids, right? I don't know if that's ringing any bells, but lipase is an enzyme that breaks down lipids, okay, it tells you, into glycerol and fatty acids. That's what they're made out of, at least for triglycerides. Lipase activity can be measured using Sudan3. I don't know what that is. Oh, it's an indicator of lipids. Sudan3 and lipids are red. So I might even put a square around that, or I might if you're doing it on a computer, have some paper where you can jot stuff down. If it's on paper, write it in the margins. Sudan3 with no lipid is light brown. Okay, so no lipid is brown. The color change can be used to measure the rate of lipid breakdown. Okay, so you would have a lipid, and it's going to break down into glycerol and fatty acids. And to do that, you need the enzyme lipase. Okay, and then what does it say? Lipids are red. So with this indicator, this stuff is going to be red, and this stuff is going to be brown. So the color change can be used to measure the rate of lipid breakdown. In the table below, Sudan3 is added to every test tube. So we have a bunch of test tubes at different pHs, and you put lipids in all of them except this one. You put lipase in all of them except this one, and you pre-treat with boiling none of them except this one. So you boil this one. So notice that you've got 4 test tubes at pH 7, but 3 of them have something else done to them. Okay. So it says justify the inclusion of test tube E as a control in the experiment. So what was E again? It's pre-treated with boiling. So when you boil, you might remember that boiling denatures proteins. Oh, here they are. Okay, so we haven't covered proteins yet, but remember that proteins are made out of amino acids. So each little, here's the thing, each little bead would represent an amino acid. And so all of the amino acids together will make the polypeptide. And their shape is super important. And so here's their shape. It's going to look like this, and it would have to be shaped just so in order to work. If you denature it, if you boil it, it's going to unravel. So the hydrogen bonds are going to break. We will go over all of this stuff again when we do the unit on organic chemistry. But if you break the hydrogen bonds, the whole thing is going to unravel, and now it doesn't work anymore. It's sort of like having a hammer that you melt, and now you can't use it to bang in nails anymore. So if you're pre-treating this one by boiling, notice you've got a test tube here, and you're putting in the lipid, so you have the reactant. You're putting in the enzyme, so you've got the enzyme, but you're boiling the enzyme, so it's now denatured. So what they had like different answers, and one of them said there's no enzyme. Not true. The enzyme is here, but the enzyme isn't functional. And so that's what they want you to see for this particular example. So these are just all different types of just examples that AP can come up with to ask about controls. They love, love, love to ask about control groups. All right, I think I've talked enough for a little bit.